Okay, so muscular system. Who has questions?
Muscular.
Or just exam? Who has questions? Either way, totally valid.
Maybe we can go over some things that'll be on the exam tomorrow so we can know about where to study.
Okay, so it's the muscular system.
Right, but like about where? Okay.
So the material that we started before spring break on the muscular system. So the
Lecture 11 through 14.
yeah, so it was lecture 11 through 14. Thank you very much. Mm-hmm. And so basically what we covered was, see if this works. And is in any way useful. This isn't working, you guys let me know. Let me go from red to black. Okay, so we covered a lot of different things, but basically everything comes under muscular system, right? So the muscular system is concerned with muscular tissue. Right, so we know that there's 3 kinds of tissue. They all have their own characteristics. We know that skeletal tissue, skeletal muscle tissue, is the one that's actually in the muscular organs, right? That we call muscles. So we know that they have a unique anatomy. So there's the anatomy of the muscle organs. There's the anatomy of the muscle cells, right? So muscle fibers. And that includes all the contractile proteins. And so once we get into the anatomy of the contractile proteins, that pretty much takes us to the physiology of contraction, right? So we need to understand how muscle contracts. And so when we start talking about it like we did, and this is typically how we teach it, we start at the level of the contractile proteins, right? And then we go out from there. So we're looking at the sliding filament model. And then that takes us to the events at the neuromuscular junction. So this is how motor neurons control muscle fibers. And then we want to know how the events, how the motor neuron actually leads to contraction. So that's excitation, contraction, coupling, right? And this is where we get calcium released into the cell. So once we get calcium released into the cell, oh, I see. Sorry, I threw a bunch of blank slides into a blank PowerPoint, but apparently we started at the end, so that's not helpful. Anyway, once we get there, hopefully we're going to see that slide again. That's when we actually get to the contraction cycle. Right, so we know that as long as there's calcium present in the cell, we're going to get this series of steps. So we're going to get cross bridge formation. And of course, you need to know what's actually happening in these steps, but this is an overview to stimulate more questions. You're going to get a power stroke, which is where we're actually moving the filaments, sliding the filaments. And then we're going to uncouple or, undo the cross bridge, lose the bond, whatever, the thick and thin filaments, so the myosin and the actin. And that's where ATP comes in. And then we're going to pop the myosin head. and that allows us to return, right? So it's a whole cycle, which means that we keep running through that as long as we have calcium present in the sarcoplasm. So that's all the like super microscopic stuff. So once we understand that level of physiology, like what's happening at the individual sarcomeres, we know that whatever happens to the sarcomeres is happening to the entire myofibril, and that each muscle fiber is packed full of myofibrils, right? And so that can translate to what happens at the level of the muscle. So then when we look at the next level of physiology, that's when we start to look at what happens to individual fibers. So now we're looking at the level of cells. And we know that when a muscle fiber is stimulated, we get a muscle twitch. So we can get one twitch per stimulation, but we know that if we keep stimulating, that we're going to get an accumulation of twitches. So that's going to lead us to more of a muscle tetany. So a twitch is a single increase in force in response to a single stimulus. And then we can get additional force as we continue to stimulate. And if we keep stimulating rapidly, we get maximum sustained force. And then we also talked a little bit about the, close enough, about the trepi. So the idea that in like an overly relaxed or like unwarmed up muscle fiber, that you'll get more tension building the first couple of times that you stimulate because they're not fully like functional yet. And we're pretty sure this is because we're actually increasing the amount of calcium being released. And so this is the equivalent of a single twitch. And these are kind of your warm-up twitches. Why am I, yeah. Okay, so that is of course all well and good, and we like to talk about that, but it's not what happens in the real muscle, right? So then we take it from the level of a single muscle fiber into what's actually functionally happening, right? So that takes us into motor units. So motor units are what we get when we have all the muscle fibers that a single neuron is controlling. And what we ultimately learned were a lot of different things, right? So we know that every fiber is controlled by one and only one motor neuron. We also know that motor neurons control more than one fiber. We know that the fibers are all going to be the same fiber type. So they might be the type 1 slow twitch. So they can be, so they can be slow oxidative, or they can be fast glycolytic, or they can be fast oxidative glycolytic. But they're all going to be the same. And we know that they're going to be distributed throughout a muscle, so they're not hanging out together in like a single fascicle or anything, they're spread out. And that single muscle organ typically has lots of different motor units, right? So when you start stimulating a whole muscle, the motor units that you stimulate first are going to be your slow oxidative. So slow oxidative motor units have, what's the word? The most excitable motor neurons. So they're the most sensitive. and they're the first recruited. And this is based on the size principle, because remember that the slow oxidative have the smallest diameter and the fast glycolytic have the largest diameter. So we always start with the smallest diameter muscle fiber motor units when we need to recruit. So if we need more force than what these guys can provide, then we're going to keep recruiting motor units based on the size principle, which is also based on muscle fiber type, until we get everybody recruited, right? So we call that multiple motor unit summation. Or spatial summation. There's a lot of names. Sure, I'm forgetting some things, but again, the details are where you guys should be looking at what you studied and saying, okay, this was something that didn't make sense. So at any point, if you have questions, just throw them out there. I lost my chat, hold on, let me find it. Don't need to keep those right now, I can always rewrite them. Hold on. There it is. Nope, sorry. Having issues. Okay. So let's see, where were we? Okay, so then what comes after that? Hold on, I got this. Okay, so that takes us from the level of, oh no, I don't want to be read. Okay, so we've got the subcellular level, right? Why would you do that? Cellular level, and then we've got motor units. So that kind of takes us to the level of the organ and the actual, oh no. I know it seems silly, but it can be really hard to talk and write at the same time. Musculoskeletal unit. Okay, so musculoskeletal unit. What that means is that, of course, muscles don't operate in a vacuum, right? So when they're actually operating in the body, we're going to have multiple muscles, and they're going to be attached across at least one joint from one bone to the other. Obviously, there's some exceptions to the muscles attached to bone rule, but for the most part, they attach to bones, and that's what we are going to concentrate on. So there's different types of contractions that we can have, right? And so there's two types generally, based on whether the muscle changes its tone or strength, or whether it changes its length. So if you have a muscle maintaining the same tone, that means that it's going to be changing its length. And there's two ways it can do that. It can lengthen or it can shorten. So when it shortens, that's concentric. And if it lengthens, Oh God, that's eccentric. Or it can maintain the same length and just change its tone or strength. Now, that sounds weird in isolation, but then when we talked about the different roles that muscles have when they work in groups, hopefully that made a little bit more sense, right? So when we look at how muscles work together, we know that for any given action, it's going to take the teamwork of multiple muscles. It's not just one muscle doing it. So the muscle that provides the majority, see now I'm combining, it's not helpful. The muscle that provides the majority of force for a particular action is called the prime mover, also called the agonist. Any muscle that helps the prime mover, either by contributing additional force to the movement, by maybe altering the movement a little bit, by stabilizing joints or bones, or anything like that, they're synergists. And then muscles that oppose antagonists, the force directed by the prime mover, are called antagonists. And hopefully it made sense that, it sounds weird that muscles are going to be acting in opposition to what a muscle is doing, but it's all about controlling and smoothing the movement. So antagonists are often going to be the guys doing the eccentric, isotonic contractions, because that's something like your triceps being active as you flex at the elbow, right? So when we go back to the whole using your biceps and triceps, your biceps brachii is going to be your prime mover when you're flexing at the elbow. You have a variety of synergists there, but your brachioradialis and your brachialis are your major ones, and then your triceps brachii is your antagonist. And that means that it's working. It's not like just relax and allowing it to happen. It's working to help control the movement. And then if we wanted to reverse the movement, so if we wanted to then extend at the elbow, then we would see a reversal of certain roles, right? So when we extend at the elbow, the prime mover is the triceps, and the antagonist is the biceps. And please don't ask about synergists, because off the top of my head, I don't know, and it also depends on what else you're doing. So, so that's where we see more of a realistic function of the muscles, because they don't actually work by themselves, they work together. That is a good question. Okay, so would I ask, would I give you a muscle and ask you to identify if it's a prime mover or whatever its role is? I don't expect you to know individual muscle roles in particular actions. So not only would I not ask you that for lecture anyways, like for lab, we're learning individual muscle names based on the list, but you don't have to know the actions. If I gave you a muscle and wanted you to know its role, it would be in a scenario. So I would be talking about, okay, so, in order to flex at the elbow, this muscle does this and this muscle does that, like then perhaps. I wasn't planning on it, but it's a great idea, but probably not. So I don't expect you to know details about particular muscles like that. Yes, exactly. So know the definitions. So you need to know that a prime mover is the one that is providing the majority of the force for a particular muscle action. And synergists can do a couple of different things. I gave you the list, but basically they're supporting the prime mover and the antagonist is opposing it. That's what I want you to know. I use the specifics as an example to try to make it more concrete, but I always use the same example because A, it's one that we're familiar with. We use it for a lot of different things, and I don't need you to know that kind of stuff about the muscles. So the last about muscle shapes. So again, with muscle shapes, you need to know like the definition of the muscle shapes, but I don't expect you to know particular muscles that fall into the categories. So yeah, like literally what you need to know about muscle shapes is that the definitions are based on the patterns of fascicle arrangement, right? So 5 muscle shapes. So we've got the circular, right? Yep, we've got the parallel. We've got the fusiform. We've got convergent, like this. And then we've got our pennate. And pennate does come in some subcategories, right? We can have unipennate, bipennate, and multipennate. 1, 2, 3, 4. Yep, that's fine. Okay. So what you need to know, you might have to be able to recognize the shape in general. You know, I could do a matching with the pictures and say, hey, you know, which one is this? But I'm not going to say which particular muscle is this, because again, that's like one step beyond. Like we don't have time for that. Like you'd have to take like a whole course on muscles for that level of detail. So yeah, and I think the last big physiology thing we talked about was our little delve into physics, right? So we talked about lever systems. So again, with lever systems, you don't need to know particular muscles or particular examples. Although I gave you examples, it was just to demonstrate how it would work in the body. What you need to know is, you know, the 3 types based on the arrangement of fulcrum, effort, and the load, and whether or not it has a mechanical advantage. And remember that when we talked about mechanical advantage, so basically what we talked about was mechanical advantage is all about the relative distance of the effort and the load from the fulcrum. So when your effort is farther from the fulcrum than the load, you're going to have a mechanical advantage. And what that means is that you have an increase in power out than in, right? So you're actually getting more power out. So it lets you, for example, move something heavier by using the lever than you would by yourself. And then the opposite is true. So then if your effort is closer to your fulcrum than your load, you're going to have a mechanical disadvantage. And that sounds bad because it means you're getting less power out than you're inputting, right? So it's actually making it harder for you to move something. But remember that there's a trade-off between power and speed. And so when something has a mechanical disadvantage, you're gaining in speed as your output. So you can have a power lever or a speed lever. And a power lever has a mechanical advantage and a speed lever has a mechanical disadvantage. And even though we would think, because of the way we're used to using levers, sort of in like real life, you know, we're used to just like using a stick to wedge a rock up, or using a seesaw, or it's mostly shovels, I think, and like hammers, I guess, not using the hammer, but the back of the hammer to wedge a nail back out again. Sorry, tangent. Anyways, in spite of the fact that you would think that power would be the desirable outcome, speed is actually more common in the human body. So almost all levers in the body are third-class levers. And by definition, a third-class lever is always a speed lever, because literally we have the fulcrum, we have the effort, and then we have the load. And that's how most muscles are attached across joints. So we go back to our biceps example. So here's our humerus, right? And then we've got a little elbow right there. And, you know, there's like a hand here. Just go with it. And so we've got our muscular effort coming in here. Here's our fulcrum. And our load is going to be back here. So obviously the distance between the effort and the fulcrum is much shorter than the distance between the load and the fulcrum. So this is a third-class lever. It has a mechanical disadvantage. So it's a speed lever. So when our muscles act, the point isn't to be powerful. The point is to just to get movement. And I think that's one of the big reasons why it's useful to teach the lever stuff when we do this muscular system information. Okay, what else? Questions? Anything that I didn't talk about at all, or that you're not sure how I'm going to ask? Or you want me to go into more detail?
Do we have to know the types of ion channels? Like open, gated?
I mean, yes. So you need to know the ion channels with respect to how they function in electrical activity. So since we talked about action potentials and how we're achieving that, and especially because, you know, voltage gated and ligand gated channels are so important here. The leak channels, I usually have trouble working into the test, to be honest, so it's probably not going to come up. But you know, none of this would happen without those guys. So it's easily as critical to know about that as to know about like the sodium potassium pump.
And then the different types of connective tissues. like the epimecium, paramecium.
Yeah.
You can,
So yeah, so that's part of the anatomy, right? So yeah, whether it's just asking questions about it or whether it's kind of knowing that anatomy, you do need to know that, because it's part of what makes muscle organs function, So yeah, I mean, if there's a section that I covered, you're going to need to know the basics of it. I obviously can't guarantee that there's going to be a question about every single thing on the test, because I only have 40 questions to work with, but I do try to cover everything. So the thing about the online testing, and I hope that, I know it wasn't very long, but I did not really have time to make a longer practice test, but I hope that having an example of each basic type of question was helpful to look at, and that it's kind of clear the basics of how this is going to go. But, basically, since we're not really on a schedule, I don't know what everybody else has going on in their lives, I'm giving you a range of time to take the test, right? So you guys will be able to access this test as early as tonight, if this evening works better. So I'm going to try to make it about a 36-hour access window. And once you access it, then you're going to have 2 hours. You're going to get every question individually. So you're going to see one question at a time. And you're not going to be able to go back to the questions, okay? So make sure that you're committing to each question before you move on. And the reason is, of course, that I now have no control over your testing environment. I'm trying to ensure as much fairness as possible. I also don't want to screw anybody over, which is why you're getting 2 hours. So I'm hoping that this balances all the factors out as best as possible. Let's see. Oh, the other thing that I want you all to understand too is that the questions are going to be sort of a random assortment from a test bank. So for every topic that I just mentioned, I'm going to put a bunch of questions into the test, and each of you individually is going to get a different group of them. So you should be getting some from each topic, but which questions you get will be different. And so technically everybody's going to get a different test. They're all going to be the same, types of questions, level of questions, all of that stuff. But they'll all be a little bit different, if that makes sense. And once the testing period is over, you'll be able to see the correct answers and see how you did. Obviously, I still have to grade the short answer part. And my other class is, of course, also taking a test. So I'm going to have a lot of grading to do this week. But as soon as those are graded, you'll be able to see those as well. So then if you have questions about the test afterwards, you can shoot me an email and we can either talk about it by email or we can set up a little Zoom meeting if you have specific questions about your exam.
So we would be able to take the test as early as tonight?
Yes.
Okay.
Yeah, if tonight works better for you than tomorrow, take it tonight. If you need more study time or just because tomorrow works better, take it tomorrow. Okay, for both lecture and lab exams. So Yeah, basically the setup is assuming that everything goes fine. This is obviously just the lecture exam that we're taking today, right? Or tomorrow or this week, whatever it is. I don't know what day it is anymore. We're doing the lecture next week, right? Next week is the lab one that I have scheduled, right? I want to give you guys one more week to access all your bone and muscle stuff. So if this all goes fine, then this will basically be the same setup for the lab exam as well, except that it'll be images of bone and muscle stuff for you to identify, as opposed to like a lot of, like it's mostly just going to be, what is this, right? It's going to be a picture of a bone or a muscle grouping with something pointing at something or highlighting something, and you'll need to identify it from a list of options. So same basic setup. And yeah, the nice thing is that I can just put like everything I would possibly want to test everybody on, and then you'll each get, 40 of those plus a couple of short answer. But I don't want to say absolutely that's how it's going to go, because I want to see how this one goes first, right? We're in this new territory, and so I want to be able to adjust if I need to make things work best for everybody. Any other questions?
Let's see, I'm not sure if you already went over this because I came in late, but did you go over tropomyosin, troponin, and actin?
I did not specifically know. Nobody asked. What would you like to know?
Just their roles. So I think I'm a little, I think tropomyosin, I'm a little bit not sure what it does.
Yeah, no, that's fine. Here, let me go out of this one and go back to that lecture so that I don't have to draw poor versions of thin filaments. On related news, it's like really raining now. Okay, that was what? Okay. Muscular tissue, scopic anatomy. Go to, there we go. That's the one I want. Okay. So yeah, it's always, tricky when we cover this stuff because like I always feel like I should teach it twice, because we cover it, you have no idea what's going on. I'm telling you what's happening, but you don't actually know what's happening yet. And then by the time you know what's happening, we've finished talking about it. And I think that makes it extra confusing, but that's kind of where, the studying comes in. Okay, so thin filaments, this is basically what we're talking about here. So thin filaments are actually composed of 3 different proteins. So the functional protein is the actin, right? And they're these little, if only they were actually green. But anyway, so they're these little globular proteins, and they have a single active site. So they have a binding site for myosin. And in resting state, the binding site for myosin is covered by this tropomyosin. So you can think of tropomyosin as these long little ropey guys that sit directly over the binding sites. And when the thin, when, hold on, I'm gonna say it in a confusing, I got this. Okay, so holding tropomyosin in place, there we go, got it, that makes more sense, is troponin. So troponin is the active guy. Troponin is like the guy in charge. Tropomyosin is just kind of there. So you can kind of think about this like the, you know, like when you're like going to a parking structure and you have to pay to get out, and then the little gate raises. So tropomyosin's like the little wooden gate that raises to let the car out, and troponin is the guy in the booth actually hitting the button, okay? So troponin actually has a binding site for, for calcium. And when there's no calcium, this is the state, okay? So when there's no calcium, troponin is positioned so that tropomyosin covers the myosin binding sites on actin. And so that's how I like to think about, this is why tropomyosin is named that, right? So the troponin is controlling it and it's covering the myosin binding sites. That's how I think about it. Sorry, if I hold it on there too long, bad things happen. So then when calcium is present, and we know how that happens, right? We've talked about how we end up with calcium, but when calcium's present, it binds to the troponin. This causes a change in configuration because that's how proteins do everything. So then the troponin basically allows the tropomyosin to move. So it's gonna rotate, and now we have exposed myosin binding sites on the actins, so that now we can bind myosin and we're gonna get cross-bridge formation. If we take the calcium away, then we go back to the resting state. So this is, again, a cyclic change, and it's entirely dependent on the presence of calcium. So they're working together, and that's why you'll see them called regulatory proteins, because they are in charge of controlling the actin.
Okay, great. That was
Any
really
other-
helpful.
Okay, any other confusion about that? Any other questions about thick and thin filaments while I'm here?
So with the components of the sarcomere, do you think you're going to put a picture and we have to identify the A band, the I band, and M line, or more know the definitions? Or?
It depends. Those are both of the ways that I have asked it in the past. So like I said, you're all getting slightly different versions of this, and I'm putting in all of the different questions, so you could get one or you could get the other. Hopefully not both if I do this right. So yeah, so sometimes I do put in the diagram and ask you to identify them. And sometimes I have the definitions, or I have it like a matching with the definitions. I've done all of those things. So, and hopefully if you're, if you're comfortable with the anatomy of it, then, and you know your definitions, then they work both ways, right? So for me, at least, I'm very visual. I like to study the diagrammatic view, and then I'm able to say, to put it into words if I need to. But it depends on what clicks for you. You might want to, be going back over the words more than the diagram.
Okay.
The same amount of free response questions, yes. Yeah, so basically they won't necessarily be the same, but they'll be the same amount, and the point value is going to be the same. So everybody's going to get 20 points worth of free response, and I'm going to try to evenly divide it up between the different like types or topics that we covered. Yeah, okay, so number of myosin cross bridges, changing the force of contraction. Okay, hold on. All of this, do not want to keep annotations. I'll go to the appropriate lecture just in case. No, that's the wrong lecture. Yep, there we go. Okay. Just have to find the right. No, I think I went past it. Hold on. Oh, there's the slide I want. Okay. Okay, so how does the number of myosin cross bridges change the force of contraction? All right, so throw a new slide in there, and then going to go to here. All right, so obviously these are all of the different factors that can increase the force of contraction. And the way they do that is every single one of them increases the number of cross bridges. So here's why that makes sense. All right, so this is where we want to go back to sarcomere first and remind ourselves that as long as the cross bridge cycle is occurring, what lecture is this in? 13? 13. It's like the third muscle lecture. Okay, so as long as there's calcium present, okay, pretend these are cross bridges, we're going to keep running through the cycle, right? Because we like never actually run out of ATP. So we're just going to keep running through and running through. And as long as we're running through, we're going to keep sliding those filaments together. And even if we run out of room to slide them, we're still going to be cycling through the cross bridge. So the cross bridge cycle occurs regardless of how much force we're actually generating. The more calcium that's present, the more cross bridges we're going to have cycling. The more cross bridges we have literally means more pulling of more sarcomeres in more muscle fibers all at once. So if you remember that whatever's happening at the sarcomere is happening to every single myofibril in every single muscle fiber and potentially in, like almost all the muscle fibers in a particular muscle, right? Because we can get total motor unit recruitment. every single cross bridge is working like a ratchet, you know, like just cranking slowly together. And it has an additive effect. So the more cross bridges we have, literally the more force we're generating, because cross bridge cycling means force generation. So anything that increases the number of cross bridges is going to increase the amount of force possible. And that takes us back to this. So every single one of these things represents more cross bridge cycling occurring at the same time. So for one thing, obviously, you know, if there's more muscle fibers, so more cells, we're going to have more cross bridges, right? Because every cell has a certain number of myofibrils and a certain number of sarcomeres. We know that an increase in muscle fiber diameter is caused solely by more myofibrils. And that, again, means more cross bridges. So both of these actually increase the number of myosins and actins that can interact with each other. And then these two things, so increasing the frequency of stimulation maximizes the amount of calcium available. And when the amount of calcium available basically frees all of the actin to bind with myosin, then that's going to increase the number of cross bridges. And then this guy, our length tension relationship, is also all about maximizing, so that every single myosin is binding in those muscle fibers. So it's basically this direct relationship where cross bridge formation equals contraction. Does that make sense? I can try a different way if that doesn't make sense.
Could you maybe spend a little more time on the wave summation and tetanus?
Sure. All right, so wave summation is about this idea of a graded muscle response. So this is where we start to see that in reality, we're not working with single muscle fibers or even single motor units. We're working with an entire muscle that's able to generate a variation of response. So whenever we have something that's able to generate a variation of response, we call it a graded response. So a graded response means that as you increase the stimulus, you're going to increase the force. So the way we like to teach this is we go, okay, well, let's look at all the different ways that we can increase the stimulus. You know, how do we stimulate more myofibrils? Well, there's a lot of different ways. So the first way is to simply send more stimulations in the same amount of time. So that's the temporal summation. So temporal summation is also called wave summation. So temporal means time. And it literally just means that we're adding stuff up over time. So this means quicker stimulus frequency. Wave summation is the descriptive term for it because it looks like we're adding up waves when we actually graph it. So basically, the idea with wave summation or temporal summation is that before we achieve full relaxation, we stimulate again. And so then subsequent twitches, instead of starting from 0, are now starting from an already, from having already generated some tension. And so it ends up adding together, okay? And so then if we keep doing that, then we keep increasing the amount of tension generated. Now there's a limit to that. We can only do it for so long because of course there is a peak level of tension that can be achieved with a certain number of cross bridges. So once you get to that point where the calcium levels in the sarcoplasm have essentially freed all the actins. So we've maximized myosin crossbridge for that particular muscle or whatever unit it is we're dealing with. That's when we end up with tetanus. So the idea with tetanus is that we're achieving peak tension, as in it doesn't matter what more stimulus you do, you cannot get any more force generated. So you've maxed out, so call it maximum cross bridges. And there's 2 ways to do it, of course. And this is one of those examples of things that happen in a lab that we don't, like I can't give you an example that's happening in the body because it doesn't really happen this way and it's nothing that we can actually perceive. But unfused tetanus is basically temporal summation to maximum force, right? Because this is temporal or wave summation right here, and it's going until we actually get to the maximum tension. Fused tetanus is when the frequency of the stimulation is so much that we never relax at all. So we just ramp the tension all the way up to max and we stay there until either we stop asking or the muscle fatigues and stops responding. And then the trepi, since it's right there on my way to going to the length tension relationship, is of course sort of this idea that overly rested muscle kind of needs a warm-up period. So basically the calcium levels in the sarcoplasm don't quite get to where they're supposed to be the first couple of rounds of stimulation. So that's why we see this increasing amount of tension from the exact same stimulus, but it only happens a couple times and then we reach appropriate calcium levels and then the same stimulus is going to get us the same amount of tension. So this is about a warm-up period, essentially. It's not really a warm-up, but that's what happens. Okay, so, and we talked about this a bit. Let's go over here to our length tension relationship. So the length tension relationship is, it's a little bit weird, but it's all about cross bridges still. So because the amount of force generated requires cross bridges, so max force is going to be achieved with the maximum amount of cross bridges. Okay, that's where we want to start with this. In order to maximize the number of cross bridges that are formed when you start contracting, you want to have all of your myosins and actins overlapped at the beginning, but you don't want them to be too far overlapped because you'll run out of sliding room. And if they're too far apart, then you don't have enough overlap at the beginning to really generate any tension. So if they're too far apart, you can't cross bridge, right? Or you only have a couple of them. It's like trying to grasp at the fingertips. You're just not going to get a good grip. And so that means it takes a little while to like, oh, finally get, ratchet everything together. And then you can start to generate more tension. Well, you just can't get to maximum that way. Not right away, at least. And again, if you're too close together already, you start generating tension, it goes really well, but then you run out of room because, you know, your thin filaments can't cross your M line because your M line is an actual physical barrier of proteins holding your thick filaments together. So if you run out of room, then you're also not going to generate maximum tension. So when they figured this out, they, you know, did studies and whatever to figure out, well, what is the sweet spot in the middle, right? What's not too overlapped at the beginning and not too stretched apart. And so that's where you get this optimum resting length. So the optimum resting length is the sweet spot where we have enough overlap of the thick and thin filaments, so to maximize our cross bridging. To maximize the cross bridges without running out of room. And we can, of course, max that, too many times. We can match that up with this idea of muscle tone, this idea that there's always a little bit of activity in our muscle fibers, regulated by the unconscious parts of our nervous system, because one of the goals of muscle tone is to maintain our sarcomeres at the optimum resting length, so that whenever our muscles are needed, they're already able to generate maximum tension. So that's where we get this graph of the length tension relationship. So this is our optimum. This is the starting point at which our sarcomeres need to be to generate maximum tension. And of course, it's right around about 100% of our sarcomere length. So it's kind of like, it's like literally resting. And we can shorten up to about 60% of the sarcomere length before we run out of room. So that's basically where the thin filaments are like hitting the M line. I assume that doesn't actually happen, but it helps me to visualize it that way. And we can also, you know, we can get some contraction when things are stretched out, right? But if we get much past this 160% of the sarcomere length, there's just no cross bridges anymore, right? Our thin filaments have been stretched so far that there's no overlap between the thick and thin filaments. And we need overlap to have cross bridges, right? Because if the actins and the myosins aren't next to one another, they can't bind together. So that's where we get this crazy looking little graph, is that here in the middle is when we can get peak tension. This is peak tension. And this is kind of our range of functional sarcomere length. And I think that, does that make sense? Is there anything else that I can say about that? Any other? Brenda, you still look concerned about it?
No, I think I got it.
Okay.
Thank you.
Mm-hmm.
What causes it to be like stretched out like that?
Bad things probably.
Oh.
When you're looking at this, I mean, I would say, I'd have to look it up to know for sure, but I would say that physiologically, like in a real life situation, we're really not straying into the bound, like the edges of this graph. You know, they like isolated muscles in a lab and put them on some sort of tension measuring meter and, you know, messed with it to find this out. Because honestly, like in reality, your other parts of your body would prevent that much stretching, if that makes sense. You know, like the fact that your muscles are connected to your bones and your bones are connected to each other and they're all bound together, Because when I start thinking about this, I start thinking about like medieval torture and putting people on a rack and like stretching them out. It's not the muscles that give, it's the joints. So really, you're not getting here in reality. All right, so yes, sarcomere, A-band versus I-band. So yeah, when a sarcomere changes length, the A-band never changes because the A-band is simply the length of the thick filaments. And because we're not actually changing the length of any of our proteins, because we're just changing the overlap, we're never changing the A-band. But because the I-band is defined as thin filament that is not overlapping thick filament, the more overlap we get, the less I-band we have, and then vice versa, right? So the more relaxed the sarcomeres are, the more I-band length we have. Any other questions? I bet there's at least one. Ah, here we go. Okay. Thank you, Ramon. The development and regeneration of muscle tissues. I can, but I did not actually cover it, and you don't need to know it. I forget to pull the learning objectives out sometimes, but if I wanted you to know it, there'd be a whole section, and there's not, so don't worry about it. Yeah, sorry. I really try to remember to edit my learning objectives, but I am copying them straight from the book, and sometimes I miss them. Yes, you need to know depolarization and repolarization. So basically all of that subcellular, all of that stuff that we talked about at the beginning, it's kind of how the rest of it works. And the other reason why I say that so emphatically is because when we're done with this system, we're moving on to, oh, it's 12, it's 12. We're moving on to the nervous system, and that's how the nervous system functions. So this is my first chance to see how, like, to get this across to you guys, how all of this electrical activity stuff works. But it's not the last time we're covering it. We're gonna tackle this stuff again in the next system. So So this goes back to your previous question about, the types of ion channels. So all of this stuff ties in. So when we talk about electrical activity, we're talking about changing the charge across the membrane. And we have two different types of tissue that do this, right? So the muscular tissue does this, but as we're going to find out literally in the next chapter, neurons do this too. So this is the way that nervous tissue functions as well. And it's very, very, very similar, to the point where the term action potential applies to both, right? So when we talk about what happens at the neuromuscular junction, the depolarization, repolarization is happening to a neuron first, and that results in the release of neurotransmitter to stimulate the action potential in the muscle. So this idea of how depolarization, repolarization, or how we get, action potentials occurs is crucial. Like, this is the physiological event that you need to understand for the muscular and nervous systems. And both of them make use of these different types of channels. Now, we don't use the mechanically gated ones right here, but we're going to come back to that when we get to the sensory stuff in the nervous system. I don't know how much time we're going to get to spend on that, considering everything, but that's where that becomes relevant. So this is basically like touch and pressure and stuff like that. So that's why I typically mention it here, but then we don't actually talk about it. The ones that are relevant for the electrical activity here between neurons and muscle cells are the leak channels, right? So the leak channels are part of establishing resting membrane potential. And that's especially the potassium leak channels. Sorry, I wrote the right thing, I said the wrong thing. And then when it comes to the ligand-gated channels, this is the events of the neuromuscular junction, right? So this is acetylcholine binding to receptors that we care about. And then everything else is voltage-gated channels. And hopefully you don't need me to go through the sodium-potassium pump again, because I feel like I've covered it thoroughly, but I'm happy to go over it again if somebody has questions about it. Of course, I'm just saying I'm skipping it right now because what I want to focus on is the actual action potential. So depolarization, repolarization are the steps of the action potential. And the idea is that it's rapid and it's a complete reversal of charge. And again, it's only occurring across the membrane. The cell itself, like the whole cell, isn't doing anything. All of the extracellular fluid isn't doing anything. This is something occurring just across the membrane. So with depolarization, we're starting with this negative charge inside the cell. A stimulus opens voltage-gated sodium channels. Sodium floods into the cell. So we have this massive positive charge influx, and that causes the depolarization. And then we obviously have to be able to reset the thing, right? So now we have depolarized the membrane, and now we're going to reset. And although the sodium-potassium pumps will reset the cell in a very slow manner, that doesn't give us an action potential. We want a very rapid reset because we did a very rapid reversal. And so that's where the repolarization comes in. So we're going to have more voltage-gated channels open. So now we're going to have a flood of potassium leaving the cell. So we get all of this positive charge now leaving the cell, and that leaves the inside of the cell negative again. And then the actual resetting event is going to be by the pumps. So we can look at this chart. This is our classic action potential chart. This is what occurs both in muscle cells and in neurons. Okay, this pretty much this exact same thing, even though the details are different. So resting membrane potential. A stimulus of some sort slightly depolarizes the membrane to threshold. That's going to trigger the voltage-gated sodium channels. And the sodium floods in, and that depolarizes us. Now, these channels are basically on timers. So the sodium channels close when we get to this peak depolarization. The potassium channels open. The same stimulus is actually triggering both the sodium and the potassium channels to open. It's just that these guys are really slow and these guys are really fast. So the sodium's open first, and then they close. By the time they have opened and closed, the potassium's finally open. Potassium floods out, repolarize, and then this is our reset. So this is, oh gosh, why am I blanking on words? This is the time when we can't, I'll just go over here. Sorry. There it is, refractory period. I'm sorry, words. Okay, so that's when we end up in the refractory period. So the refractory period is when we're non-responsive to a stimulus. We can't cause another action potential because we're resetting resting conditions. Okay, so this is important enough that I break down the basic events of how that happens, and then I give you the specifics of what's occurring because it's happening at the neuromuscular junction, and we're triggering the entire muscle cell depolarization, repolarization, or muscle cell action potential by sending the acetylcholine across this synapse. I was not planning on doing one because I don't have anything new to add. But if anybody has lab questions, we can definitely tackle it then. But really what we're doing this week should be the same thing that you guys were doing last week. So I can pause this or stop the recording, and then the exam, I was gonna, let me see when I put it up. I want to say something like 6 p.m. What did I do? Yeah, I think it's 6. I can just make it 6. I can always fix these things. Just make a note, make sure it's 6 p.m. All right, any other questions about lecture, muscular system, lecture exam? If you haven't done them yet, you don't have to complete them. I can extend the due dates. I was just picking a week to make sure everybody started looking at the McGraw-Hill stuff, but the due dates honestly aren't important for that stuff. So as long as you're done with that before the lab exam. So I'll just push those back to next week. Don't stress out about that today.
Are we gonna have lab exam? No.
Sorry, say all of that again.
Are we gonna have another review for the lab?
We sure can. Yeah, we can do that on next Monday, right? That makes sense. Because we've got, where is it? Lab exam. Yeah, we can do the why don't we do the review on Wednesday at 10?
Sounds good.
Okay. Oh. So yeah, after lecture on Wednesday, we will use our lab time to do a review of the lab materials. Okay. Okay, hold on. April, when we're done with an exam and receive a grade, the short answers. So the short answer stuff won't be up right away. So but they're going to be part of the grade, or it'd be silly for me to ask it. But whatever you get back initially is just going to be the multiple choice portion. And then I will still have to grade the short answer stuff. And then you'll get your total grade. Does that make sense? So it's the same thing that we did before as far as points distribution and type of questions. It's just that online means you're going to see some of it earlier than you would have anyway. Okay, good. Okay, let me